Hello, I'm Ushma Neill, here today with Vincent Racaniello, a poliovirologist from Columbia University. Vincent has also taken on the mantle of Earth's virology professor. Beyond the classes he teaches at Columbia, he also runs a popular website called This Week in Virology, where he talks about interesting and newsworthy viruses and their place in today's world. Vincent, I'm so glad to have you here today.
Thanks, Ushma.
Do you think you could tell me a little bit about This Week in Virology?
We do a podcast every week. We get a bunch of virologists together and we just talk. And there's no script. We pick a couple of articles or what's going on in the news. And the idea is to hear virologists talking about their field in a way that a lot of people can understand it.
Do you have a favorite virus?
It would have to be the virus I spent over 30 years working on, which is poliovirus, which is about to be eradicated, which is good for the world, but sad for me in my laboratory, I guess.
Are there other viruses that interest you?
Well, I think the viruses that are basically in every cubic inch of the world are amazing, in the air, and all the animals and plants and insects in the world, and in our oceans. I find it amazing that they're all around us. You can't see them. Their mass, if you put all these viruses together, would weigh 1,000 times more than all the elephants on the earth. If you line them up end to end, they would stretch 200 million light years, way past the nearest galaxy. So these are things you can't see with the naked eye. And as we go about our daily lives, we can't see these things. Yet they have enormous impact on Earth. And probably if you took these viruses away, life would be very different. I would venture to say that we probably wouldn't exist if they weren't here.
There's probably any number of things that I haven't previously appreciated about viruses. Are there any particular factoids you could enlighten me with?
Well, viruses constitute about 8% of your and my and every other human on Earth's nucleic acid. And that 8% got in many, many years ago because of a series of infections. And some of that is actually useful to us right now. Some of those bits of viral genes contribute to the formation of the placenta, for example. So if it weren't for viruses, we'd be laying eggs. And they would probably be blue, because we know that some viruses contribute to eggshell color in chickens. There's also one of that 8% that makes our stem cells able to differentiate into all the other cells in our body. So if we have during our evolution acquired these infections, and in some cases we use them.
You know, you also mentioned something in passing about the sheer number of viruses that we excrete every day.
So each of us humans, not just humans, we have at least a dozen viruses, different kinds of viruses in us at any given time. We get infected shortly after birth or maybe within the first year of life, herpes viruses of all sorts, and those viruses stay with us. That's their modus operandus. Periodically, they produce more offspring and those infect other people, but for the most part, we are okay with that. But even beyond that, we have viruses in our bloodstream, tiny, tiny viruses with DNA nucleic acids that are in everyone, and we have no idea why they're there. And here is my favorite factoid about viruses in people. Each of us excrete in our urine every day about 100,000 viruses per milliliter. Per milliliter. So next time you go in a public bathroom and you flush the toilet, which of course generates an aerosol, just remember you're inhaling someone else's viruses.
What do you hope to accomplish with This Week in Virology?
So I'm hoping that This Week in Virology reaches enough people to teach them all about how cool viruses are, how they are an amazing part of the fabric of what we are, and that most of them are harmless. For example, if you took a liter of seawater, that liter, anywhere in the world of seawater, would have more virus particles than people on the planet. But if you drank it, you'd be fine. Those viruses make a huge contribution to all of the food webs in the ocean, to photosynthesis, to cycling of carbon and nitrogen and phosphorus, but they're harmless to us. I want people to realize how many good things arise from viruses, and that the ones that make us sick, the influenzas, the noroviruses, the chickenpox, those are a minority. I view those in a way as accidental infections that are probably not meant to be, because most of the virus infections out there are beneficial. So I want to teach this to people. I want them to understand viruses. I'm hoping along the way that they'll understand that vaccination is a good thing and they won't be afraid when they hear the next outbreak. Someone out there make me a virus particle. Though this happens to be poliovirus, but it's much bigger than it really is, of course. But it just shows you the simplicity how these particles are built. So that's basically a virus. In this case, it's a shell made of proteins. And inside the shell, you can just see sticking out here is the nucleic acid or the genetic information. And this person beaded for me the nucleic acid of poliovirus. So there are 4 different bases making up the viral RNA. There's a U, an A, a C, and a G. So she gave them each a different color bead and strung them together in the right order. And this bead, which I guess I could call it a necklace, is 7,440 nucleotides long. And when this virus infects a cell, so this virus, in order to make more of it, has to get inside of a cell, and this nucleic acid has to come out. And you see, it's taking me a long time to do this. This is 10 meters long, and I'm getting stuck. Can you imagine? This happens within seconds, probably microseconds in a cell. It's all worked out beautifully. The chemistry is just gorgeous. Should I take it all out? See how long it takes. So now we're getting tangled. Oh, so she's put magnets in here to delineate regions. But this is a process we call uncoating. This nucleic acid has to get out so that it can be made into protein, so we can make more virus particles. We're almost at the end here. So in human time, you see this just takes forever. And now this nucleic acid would be out in the cell. It would go and direct the synthesis of new particles. And this wonderful vehicle, this beautiful, what we call a capsid, this would just be degraded and fall apart. Its purpose is finished.
I'd love to hear what your opinion is on Ebola, which is really in the news right now in 2014 after sickening and killing hundreds and thousands of Africans, and even more specifically about the controversy of flying back to Americans that were sickened with Ebola and whether or not that was a good idea.
Sure. So I want to preface it by telling you a story about what got me interested in being a virologist. When I graduated from college, I wasn't sure what I wanted to do. So after college, I went home and I got a job as a lab technician. And during that year, I read a book called Fever by John Fuller. And fever was the description of the first emerging virus infection. It was Lassa virus, which emerged in Nigeria out of nowhere and started killing healthcare workers, very much like Ebola does. And that book made me want to be a virologist. A lot of that story actually took place at Columbia University Medical Center where I work. So fast forward now, we know there are other dangerous viruses which have animal reservoirs. And in places like Africa, they can inhabit bats or other animals in the forest. And where bushmeat is used as a source of food, people can get infected inadvertently with these viruses. So that's what happens with Ebola. You get some meat that has the virus in it, you eat it, and then you get infected. And unfortunately, the virus is not well adapted to people. It doesn't play nice with them. So it causes a very, very nasty hemorrhagic disease, which is often fatal. Now, the problem in the countries in Africa where this disease is spreading is that the healthcare facilities are not well developed. So here in the US, we have something called infection control. If you're in the hospital and you have an infectious disease that is likely to spread to others, you can be very well isolated and it won't infect anyone else. Unfortunately, if you have Ebola hemorrhagic fever in certain places in Africa, you go to the local hospital, which may consist of a room or two. You're likely to spread the infection to the healthcare workers who are taking care of you. They'll then go home and spread it to their family. If you die, your family will prepare your body in the traditional method. They'll have to process it themselves with their own hands, and they will get the infection as well. It's nothing at all like influenza, where if I had flu, you would be getting it, of course, but Ebola doesn't do that. So bringing a person back from Africa with Ebola hemorrhagic fever is absolutely fine. That person will be put into a high containment care facility. I believe they're going to Atlanta. And healthcare workers will exercise exquisite infection control when they deal with the patient, and it will not spread to anyone else. So it's actually a good idea because those individuals will not be able to spread the infection to anyone else. Of course, if we could bring all the infected people back, it could stop the outbreak, but we can't do that. So it's a great idea, and people should not be afraid of it.
Can you leave us with one final parting thought about how interesting virology is?
Virology is so interesting that I think viruses were probably on Earth before cellular life as we know it. I think there were little bits of these nucleic acids floating in the primordial soups of the developing Earth, and from those emerged cellular life. So I like to say that all of us came from viruses. If you look at it that way, they are really cool.
Thanks for joining us here on The Wild Types, and thank you again to my colleague Vincent Racaniello. I think we'll all remember a lot of these little things about viruses, and I have a feeling you'll never look at your pee the same way again.